to bear with us, but certainly for this first lecture. And Matthew is working on aspects of specialization and diversification and plant-insect relations. And the nice thing is that he has also worked a lot on the monitoring data that have been available from the region of California to assess trends in butterflies. So Matt is really well placed to offer well, an American perspective on the future of butterflies, and perhaps have well, add new insights from a different continent. So Matt, I'd like to invite you to share your screen, and well, the screen is yours.
Great, thank you. Is that good? Can you see that?
Yes, that works fine.
Great. Well, it's a real pleasure to be able to talk to all of you, and thanks for the invitation. Of course, it would be more fun to be there in person, but even virtually, it's a pleasure to be part of a community of people that care about butterflies. So that picture there in the slide is to remind me to say that this will mostly be a Western American perspective on butterfly conservation. That's a place we go on the western edge of the Great Basin for plants and butterflies that we work with, and it's an arid place. You can see a dust storm kicking up in the distance there. And it's landscapes like that that I want you to have in mind when I'm talking about Western butterflies and these questions. So these are the questions that I've been after for some time now, I guess more something like 15 years really. So what's the status of Western North American butterflies? Can we understand or predict which species are in more severe decline? And where on the landscape do we see the most severe declines? All these questions have conservation implications, of course, particularly the last one maybe, which I think you'll see. But let's start with that first question. And a priori, you might think that this region would be a place with relatively stable butterfly populations. After all, we have really enormous areas of undeveloped land. It's quite easy to get away from cities and agriculture. We have a lot of topographic and habitat diversity. We have hundreds of mountain ranges just in Nevada, where I live. We also have large protected, legally protected areas, but it's a big region, and these are the three very different data sets that I've been using to try to understand what butterflies are doing in the West. So the Shapiro transect from Northern California, that's the data set that I've been working with the longest. So there's 10 sites there from Northern California that have been monitored every 2 weeks during the butterfly flight season for what will now be, it's very exciting, this spring will be 50 years at the first sites that were started by Art Shapiro from UC Davis. And Art still collects data at the 5 lowest sites. And then out of my lab, we've started collecting data at the mountain sites. Then in the middle there, you have locations where the North American Butterfly Association organizes volunteers to go out every summer and do butterfly counts. The color for these locations are for elevation, just so you can see some of that variation. So these are once a year counts. We call them 4th of July counts, even though they're not necessarily on the 4th of July, and sometimes they're more than once a summer. That's the data I've been working with the most over the last year, really. That's sort of been my pandemic project. And then you see iNaturalist records on the right there. iNaturalist records have great spatial coverage, but they're not really designed for time series analysis, but we've found a way to use them in that way. And to cut right to the chase here for the first question, in these 3 very different data sets, experts, volunteers, different temporal density of observations, we see a majority of butterflies in some kind of downward trajectory. So that's what's being tallied up here with the histograms. The gray bars count up butterflies that have negative year coefficients. So sometimes slight, sometimes severe. 79% of the butterflies in California have that negative downward trajectory, and then you see the fractions there, the percents for the other two data sets. We've known for many years now, almost 20 years, that butterflies at low elevations in California, so starting way over here, were in pretty severe decline since the 90s, and I'll show you that in a bit. Only in the last 5 or 6 years have we realized that butterflies in the mountains in California are also going downward in terms of density. And that observation really inspired us to go out and get these other data sets to figure out what butterflies are doing across the broader region, because the possibility that butterflies are declining in the relatively pristine Sierra Nevada seemed quite serious. So we wanted to see what was going on elsewhere. It's not good news, obviously. Looking now just at that middle data set there, the NABA data, we used that data to estimate a rate of change for just the total numbers of butterflies observed. So forgetting about individual species, which is a nice way to look at this citizen scientist generated data, just the number of individual butterflies people see, we estimate 1.6% fewer butterflies per year, which is a fairly shocking number when you compound that annually. To make it more concrete, if you imagine going to a nice mid-elevation meadow in the mountains 20 years ago, you might have seen 1,000 individual butterflies. Now our model predicts that if you go back to that same meadow this year, you would see about 725 individual butterflies, so more than a quarter loss. Let me give you a couple examples now of individual species. Here's one that's still kind of a shocker to me. So I grew up in California, and this is an animal that in the mountains you used to it was just a part of summer. So the West Coast lady, you see its geographic range there from iNaturalist data. It pretty much encompasses the entire West, which is why it's sort of symbolic of everything that's going on to me. So this is a large, robust, dispersive animal. In addition to native hosts, it eats a common mallow, which is actually a European herbaceous mallow that's a weed here. It's a very robust plant. It's like grows in every sidewalk crack. The butterfly used to, be found using it in many places, and now this animal is in very steady decline everywhere. It's still surprising to me to see these trends. So this is averaged across locations by year within these 3 data sets and put on a standardized scale just for visualization. Here's an animal with totally different biology for a butterfly. So this is a lycaenid, less dispersive, known for localized ecotypes, also in pretty severe decline. Those 2 species are among our top 50 butterflies that we rank from the most severely declining species. What you see here are results from hierarchical Bayesian models, where for each species, we estimate a rate of change or a population growth rate for locations, which are the lighter dots there, and then for the whole species, which is in black. And then anything to the left of the vertical dotted line is something with a population growth rate less than 1, which is to say in some kind of decline. You can think of that as a traditional ecological lambda. There's our monarch, which I always have in here when I talk to North American audiences, because it's our kind of standard benchmark, right? We think a lot about the monarch these days. The public knows the monarch. People are always surprised to hear that it is not the worst off butterfly that we have, even though it's quite dire in the West these days for the monarch. It's just among a crowd of butterflies that are declining. And so if we think about trying to understand, natural history traits, et cetera, that predict rates of decline, you can see right here it's going to be pretty challenging because there's a lot of variation in biology going into this top list. So we've got these 50 top decliners. We've got another 200 species that we have this kind of data for. First question, well, the second question that I'm telling you about today is, can we understand something about the biology of these species that are in decline? We did analyses that many of you have done, taking natural history traits, body size, specialization, geographic range, and asking if they are associated in a multiple regression context with rate of decline. And we didn't explain a lot of variation. So it's multivoltine species are doing a little better. So from a multiple regression type model, there's an 88% chance that species with more generations per year are in slightly less severe decline, and species that are overall more abundant. these models were not terribly satisfying, explaining much less than 10% of the variation. Volcanism is still interesting though. Even though it's a pretty subtle effect here, over on the right is from multiple regression models where we're accounting for other variables. On the left is just the raw data. So you have growth rate on the y-axis, and those means are different, but it's not very impressive, right? But I will come back to volcanism because it's intriguing to me. And I think part of the reason these models don't explain so much variation is that we are encompassing such dramatic habitat variation across these mountains. I'll come back to that. But for now, let's move on to this third question, because if we can't predict where species are declining, maybe if we can't predict which species are declining, maybe we can understand where on the landscape they're in more severe decline. Turning still to or sticking with the NABA data, here is that data shown in a slightly different way. So here the dots are rates of change for total numbers of butterflies seen, but for individual sites. So these are 72 focal sites where we had at least 10 years of data. And so it's rates of change, kind of like lambda, but for all butterflies at a site. And you can see they're mostly clustered to the left of 1 because there is this overall decline. They're ordered here by latitude and colored by elevation, but it doesn't matter too much because there isn't a simple latitudinal or elevational pattern here, which raises the question of what variables might predict this variation among sites. So we investigated this with static descriptions of climate. I mean, 30-year normals, rates of change in climate variables. So indices of climate change by season, which ends up being interesting. We also looked at the fraction of surrounding land that is agricultural or developed land. We also looked at rate of change in land use. That didn't end up making it into final models, and we tried a couple other things that I'm not going to tell you about. So we used Bayesian ridge regressions that let us take a whole bunch of correlated variables and estimate coefficients. What rose to the top was surprising at first, but I think it makes a lot of sense in retrospect, which I'll hopefully convince you of. So what you see at the top here of our variable list was fall maximum temperatures, rate of change in fall maximum temperatures. So warming in the fall, warming in the summer, and then overall just precipitation, wetness of these locations. What's interesting and complicated is that we get contrasting effects of these two climate change variables. Now, the key thing to remember, which I admit I didn't fully appreciate until this last year working with this data, is warming in a climate change context is not homogeneous either among seasons or across the landscape. Some locations warm more in one season than another. Now, I think the positive effect of summer warming, I think, is partly methodological in this data, in that if a July day you get volunteers out to count butterflies, is a little bit warmer, their ectotherms are more active, you counted more while accounting for these other variables. What's more interesting to me is the negative effect we observe of fall maximum temperatures. And there's a few hypotheses here that I'm sure are probably popping into people's minds. One is plant stress. So these are arid ecosystems, that landscape that's still in the background there, where most of the precipitation falls in the cool months. So by the time you get to the end of the summer and you're transitioning into fall, it's as if the ecosystem is kind of holding its breath, waiting for the first precipitation. And so at that point in the season, if you warm it, I think it's very easy to imagine stressed host plants, stressed nectar plants having an effect on butterfly populations. Other things are possible too. It could be that warming temperatures are interfering with overwintering cues, etc. More things that we have to learn for sure. And I'm going to show you one brand new result that isn't published on that in a minute. Just so one other thing I wanted to say here, this analysis also accounted for spatial autocorrelation. That's what these MEMs are, Moran's eigenvector maps. Is getting late in the evening for multiple syllables. And the coefficients here are not that easy to interpret. They're in units of standard deviation, and they're well controlling for everything else in the model. But these effects are strong enough that we can even eyeball them with the raw data. So here you have rate of change in butterfly density plotted against the difference between fall and summer warming. So in the lower right quadrant, places that are warming more in the fall have fewer butterflies observed. So this is interesting to me because speaking for myself, and I think it's probably true of other folks thinking about climate change, we've really thought a lot about spring and changing phenology and potential mismatch. And I think we've thought about that because that's where some of our first climate change observations came from. But it's possible that fall warming is really quite detrimental for these animals, maybe especially in the arid area, but maybe not. It may be something that we should be thinking about. Now, does this mean that climate change is the only challenge facing these butterflies? No, of course not. The thing to remember, and that I'm happy to have a chance to say, is that these sites, which you see the dots there, these are sites that were picked by butterfly enthusiasts to be nice places to go look for butterflies. And as I said in the beginning, it's relatively easy where we are to find pristine areas that are somewhat removed from cities and agriculture. Here you see histograms summarizing the fraction of land around these focal sites that was either urbanized or agricultural. Most sites are relatively pristine, which is to say this study was focused on natural areas and the impacts of climate change. It doesn't say that urban and pesticides and other things are not important. Okay, but still thinking about climate change now, my hypothesis is that climate change is really having a broad impact across the West. So let's take a look at how climate is changing across the continent. This was rather surprising to me when I first saw this graph. So this was from a paper a few years ago where these folks wanted to ask if climate change was disproportionately affecting national parks. Most of the national parks are in the West and in Alaska, and that is where this continent is really heating up. The West is just cooking at a rate that the East is not. And here you could, so that's temperatures rising. Here, over here you see drying out. So the sort of salmon colors there are rates of drying, which are mostly clustered in the West. So the drying and the warming West. So if my hypothesis is right, my hypothesis is that climate change is negatively impacting butterflies in the West, then I should be able to make the prediction that butterflies in the East are doing relatively well. You know, admittedly, I don't have a lot of degrees of freedom there. We didn't do analyses of the butterflies in the East, but luckily this great paper just came out within the last couple of weeks where Crossley and company took one of our data sets that we were also working with, the NABA data, but looked at it across the whole country. Here's what they found for rates of change in butterfly populations across the whole country. They also find that the West is a giant hotspot for declining butterflies, whereas in the East, they're doing relatively well, at least relative to the West. So I'm convinced that climate is really warming, temperatures are really impacting butterflies in this country in a massive way, focused across the whole arid West. But as you probably all know, when you talk to land managers and conservationists, they still want to know, but which is more important, and when is land use versus pesticides versus climate the most important? To attempt to answer that question as I approach an end here, let me zoom back to the Shapiro data, which is the data set I've worked with the longest. And it's the best for this area in a way because it's the most temporally intensive and we know a lot about the butterflies at these sites. And it has this elevational gradient where we have 5 sites that are very close to development and land change and pesticides, and 5 sites that are relatively removed in the mountains. And what's interesting is that those butterflies have entered into decline at different times in those 2 regions. There's a lot going on in this graph. Apologies for that, but we don't need all of it. The low elevation sites are summarized with the hotter colors, and the colors match the elevational profile over here. The cooler colors are the mountains. The interesting thing is that the declines at low elevations have been clustered in previous decades. So the 90s in particular were bad for the low elevation butterflies. And we've done previous work to suggest that that was driven by land conversion and pesticides being more important than warming conditions for that loss. Butterflies at low elevations have kind of stabilized in recent years, whereas in the mountains, we have a dramatic downturn that we hadn't actually really noticed until these mega drought years. So we had a 1,000-year drought, which is that bracket right there, which really alerted us to the fact that the mountains were drying out And note that the depth of decline, oh, sorry, I should have explained the y-axis here. These are these standardized population indices averaged across all species at each site. So it's fairly crude, but it gives us the patterns. Note that the depth of decline in the mountains removed from development is nearly half a standard deviation away from the long-term average, which is to say it's reaching what the valley had already reached in response to pesticides and land use change, et cetera. So really sort of striking pattern here. We think this is climate change up top here. We still have a lot to learn, of course, and we would like to learn, and here's where I'm going to give you just a tidbit of a recent result. We'd like to know if that climate change impact in the mountains is more mediated through plants, as I mentioned before, or direct physiological stress on adults and juveniles. It's a big question, right? It's something that we've all thought about. Kind of thinking about experiments to do in the future, but for the moment, what we're working on right now is bringing in a new layer of data, being satellite-based imagery about plants from which we can generate greenness indices, NDVI and DWI. So specifically, we're using Landsat imagery right now, just two snapshots here. Here's our big meadow at our highest mountain site, Castle Peak, very dry in the drought years, green after the drought. We're using data like this from different seasons, from many years. We have decades to build models like this. And apologies, I know I should be winding up here in a bit, and this looks complicated, but there's one interesting punchline I want to give you. So models like this, we have weather in the previous year affecting plants in this year. We have weather in this year affecting plants in this year. And then plants in this year can both affect butterflies this year, sort of see more butterflies because there's more nectar and the caterpillars that spring did better. Plants in this year can also affect butterflies the next year if, for example, the eggs that hatch in this year have more food to feed on, they do better overwintering, and we see more butterflies that next year. So we can compare the plant effects to direct what might represent physiological stress, climate factors directly pointing to butterflies in the 2 years. And just from one site, because this is just brand new results, we get a pretty intriguingly simple signal of snow in the current year, more snowpack leading to greener plants, leading to more butterflies the next year. So I think it's that effect of better food for the caterpillars that hatch that year, produce more butterflies the next year, overwhelmingly stronger than any signal of direct sort of climate stress on the butterflies that are observed in that year or the next year. Interesting in a climate change context because it suggests it's plants, which, you know, raises questions about the experiments we should be doing in terms of thermal performance curves. They may not be as important in this landscape as what's happening to plants. And with that, let me wrap up here because I'm watching the time. Are declines widespread among Western butterflies? Yes, there's little doubt on this question. I did want to mention one very quickly power analysis we did. So we took our 72 locations and we did sort of a brute force power analysis. The y-axis here is our probability of an effect, and I drew a line at 0.9, 90% probability of being in decline. We found that we needed 50 or more of those sites to be able to detect a regional decline, which I think is something interesting to keep in mind when in the literature we see, you know, some studies find a decline, then some studies don't. It really takes a lot of sites, as I'm sure all of our intuition tells us, to detect declines across a big region. Can we understand which species are in more severe decline? We're working on this, but we are producing prioritized lists for conservation groups. And where do we see the most severe declines? Places that are warming and drying the most are the worst for butterflies at the moment. They are as bad as agricultural and urban areas, which we already knew about. We hope that this can provide some guidance for restoration efforts in the coming years. And there is a lot of interest in that in the moment for right now in the US associated with pollinators and the monarch, et cetera. And with that, I apologize, I went a little bit over. Thanks to all these folks, and I put citations there for two of the things that I talked about the most. And also there's the email. So as mentioned, it's late here and it's almost 1 o'clock and I actually do have to teach at 9 in the morning. So I'm gonna make it through a couple talks, but I might not make it to the breakout session. But if anybody has thoughts or would like to chat, whatever, there's my email address. And maybe there's time for one question.
Yes, sure. Yeah, thank you very much, Matt. This is a fascinating fascinating talk and well, wonderful insights from a new type of or another type of environment. And these climate change effects seem really well, important and also interesting. Just for clarification, the fall warming impact was from the year preceding the counts, right?
So in the main analyses, the ones that we just published, locations that have been warming more in the fall have fewer butterflies.
Yeah.
So that was not a time series analysis per se. So there wasn't a lag in that, if that makes sense, right? Now we're working on time series analysis to ask about whether or not that effect is manifest through the current year or the next year.
Okay, yes.
Yeah.
And did you see any differences depending on species traits in that fall warming effect?
No, it seems remarkably uniform. Oh, but thank you, because the one thing that I didn't mention that I meant to get back to is volcanism is really interesting in our region. It seems to have different effects at low and at higher elevations. At low elevations, there's a clear positive effect. Multiple generations means you actually can do well in some warmer and drier years. Whereas you go up in the mountain, the multivoltine species are doing worse, and we see a flip, and the univoltine species do better. So there's some interesting complexity there that we need to get into.
And it might also be that, well, we see in general here that the volcanism also is related to the hibernation stage.
Yep.
So that might also be a sort of interaction.
Yep. Agreed. And that probably has elevation specific effects too.
Yeah. And there was also a question about, well, whether the presentation on nitrogen, I don't know if you followed it yesterday, but of course, well, nitrogen deposition is a big issue, but is also being studied in the US, of course. And especially in the Central Valley, you would expect big effects of nitrogen potentially. Have you made any analysis on it?
I really should look at that, and I just admit it's one of those angles of data that we have not looked at yet. I've had the feeling that it wouldn't be as important in California because it's so arid, and I feel like the sort of the limitation of precipitation might mean plants don't take advantage of it as much as in some parts of Europe, but I don't know if that's true, and it's something we should look at.
Yes, and we have a lot of very poor soils, but of course in the Sierra with the granitic soils, you might also have similar influences.
It's possible.
Yeah. Okay. Thank you, Matt. I think we have to leave it at that, but it was a really fascinating talk, and well, perhaps a last question, if you have any, well, recommendations to us on how well we could incorporate different approaches in our butterfly work.
No, I would not be so bold as to make any recommendations at 1 a.m. for me, but thank you.
Okay. Well, but we'll be in touch for sure. And thanks a lot for being with us.
Yep.
Okay. Then we'll move on to the next presentation, and that will be given by Jacqueline Loos, and she has been working in very nice regions in Romania on butterflies, and has also been getting into the topic of farmland biodiversity. And Jacqueline is now a professor in sustainable use of natural resources at Leuphana University in Lüneburg. And she will take us to the social-ecological dimension of butterfly conservation. And so, Jacqueline, I would like you to invite you to share your screen. It's in presentation mode right now. So if you can, was in a different mode. So can you, we don't see your presentation yet.
Okay, I'll start again.
Yes, and then the full screen. Okay, yes, that's a common problem.
Still not working?
No, because we you apparently didn't click off your the presentation.
Okay, it's interesting. It worked last time, so
yes.
I'll try it one more time. How is it now?
Yes, that works better. Yes. Okay. Thank you. Please go ahead.
Okay. So first of all, thank you very much for having me for this invitation. And in complementation to the presentations yesterday, where Michiel, you have asked whether we are able to halt the biodiversity decline, I would like to expand today on how we may be able to increase conservation effectiveness or conservation success for butterflies through not only engaging people, but actually to widen the perspective on social ecological systems.
Just one more point. Manon is the moderator, so please address your questions to her. Okay, sorry. Thank you.
Thank you. So yes, we have heard a lot negative news about biodiversity decline. We heard about a term used, insect Armageddon. Yesterday, we heard that over the past 30 years in the Netherlands, 50% of all butterflies have been declining. And that would mean if we carry on this trend that when I retire in 2050, there will probably be very few butterflies left if we keep on this business as usual. However, we have also heard by Frans Wassen that the EU has come up with a strategy to protect 30% of land for protected areas, so that with an increased conservation efforts, Leclerc et al. Have predicted that we may find a leveling of the biodiversity loss. However, the state of biodiversity is expected to still be lower than we find nowadays. Moreover, the effectiveness of protected areas is limited, as many examples across the world show. This may be true because in many cases we don't have enough staff, we don't have the good knowledge about timing of different management interventions, and also conserving land through protected areas can also be quite a costly endeavor, which may decrease the acceptability of an ever-growing population of humans. So if we stop thinking only in terms of habitat and matrix and rather move towards an integrative approach of conservation and consider land also outside of protected areas through integrative approaches, then we may be able to integrate conservation, restoration, and sustainable use to bend the curve of biodiversity loss and create an environment in which species may survive and thrive. So when we talk about conservation, we often talk about conservation of nature. And I find this view on nature a bit limited, as the majority of land, in fact, more than 70% of our terrestrial surface has already been imprinted by humans. So only looking into protected areas may not be the most effective approach to conserving land, especially since we find some evidence and some examples, especially from cultural landscapes, which show that these landscapes can host biodiversity and have in fact increased the heterogeneity of our environment. However, conservation and land use are often still considered as mutually exclusive, even though they are in fact part of the same system of a social ecological And characteristic for any system, be it a simple or a more complex system, is that it consists of various components. And each component in itself may only show part of the entirety, such as illustrated here by this parable of the blind man and the elephant, where each different person feels a part of the element and derives a conclusion on what this specific component is. So we see that somebody touching the tail, the trunk of the elephant here says, this is a snake. So one perspective only on system may not be enough to grasp the entirety. And Merckx has very nicely defined what social ecological systems are. He said that these are integrated, complex, adaptive systems in which social and ecological subsystems are coupled and interdependent so that each is a function of the other, which is expressed in a series of mutual feedback relationships. And in conservation, we often focus on these specific relationships. We argue that biodiversity is to be conserved because this has an impact on ecosystem functions, which eventually determine the ecosystem services that provide benefits to humans and ensures our well-being. So this is arguably quite an instrumental view on the values of our winged six-legged neighbors on Earth, but so is the rather mechanic view on social ecological systems as it has been developed by Nobel Prize winner Elinor Ostrom. And Elinor Ostrom categorized social ecological systems, such as I display here in the figure of a butterfly, as a system that is embedded in a specific social, economic, and political setting, and which is related to specific ecosystem conditions. Now, social ecological system entails a resource system that imagine, for example, a forest, which is called the resource system. And this resource system hosts resource units, which could be single trees in this case. And these are used by different stakeholders or, as outlined here, as users. The use and the caretaking of these resources in the system is derived and navigated by a specific governance system. So all of these components of the system, whether we think of a meadow, a bog, any piece of habitat, they interact with each other. And in their interaction, these components of the system determine the fate or the specific outcome, both for, say, the trend of butterfly populations, but also for people's advantages or disadvantages of these interacting system. Now it can get a bit more complex even when we consider that there are non-linear processes active in social ecological systems. For example, when we think of temporal and spatial dynamics, such as Matt has just illustrated in his presentation. So in complex adaptive system thinking, we distinguish 4 phases through which systems can evolve. These phases can also be observed in nature, for example, when we think of butterfly populations, fluctuations. So typically we find that there is a growth phase, which you can see in this field of this lying age. And this growth will carry on in nature until, or very often until, the carrying capacity of the environment is reached, such as illustrated here by this K. This is often the quadrant where we consider our conservation action to be most successful. But populations and also entire systems can also enter stages of release and a decrease afterwards, and thereby undergo circles of reorganization and restoration. So my main message here is that our conservation targets may be broadened to allow systems to undergo its natural dynamics rather than sticking only to an ideal state here in the right top corner. So this means that we also need to accompany the systems over longer term periods. One approach to understand the dynamics of these social ecological interactions, which I illustrate here for an example in South Africa on human-wildlife relationships, is using causal loop diagrams. This may help to derive insights on reinforcing or detrimental relations. For example, in how far people can use caterpillars as protein source for increased food security, or in how far butterflies are perceived as disturbance to farming because caterpillars may feed on cabbage. Or it may help us to understand why people are motivated to plant flower strips to support the nectar provision for butterflies. So conservation, in contrast to my earlier slide, is, as seen by people, not only a means to an end, but in fact, people value butterflies for non-instrumental reasons as well. In our interviews with farmers in Romania and in South Africa, we found that many people value butterflies for intrinsic reasons, and many people were also indifferent towards butterflies. And this indifference only held true as long as there were no negative consequences expected from conservation. But as soon as farmers would fear that their access to the resource or their freedom in decision-making were threatened, then this attitude may have changed. So decision-making plays an important role in conservation at different societal scales. And for the farmers that we talked to in South African context, decision-making was determined by different narratives that that motivated people. But first, I'd like to say that Matt Koloff et al. Have defined the decision-making context to consist of a set of values, rules, and knowledge that may eventually impact the decision-making of farmers. And I'd like to add here the dimension of power to show that it's not only knowledge and not only a value that eventually leads to a specific environmentally friendly behavior. So in our example in South Africa, we found that some of the most endangered habitat in the Swartland, which is Renosterveld, was occurring on farmers' field, and many farmers treated this land as wasteland, whereas others would see a value behind it and therefore keep this land for conservation issues. However, we found that those people that were treating this habitat in a pro-conservation manner, they would not stick to formal rules, but rather allocate or identify the behavior towards informal rules, and they would use their local ecological knowledge to work with these habitats. Whereas there was also another conservation narrative in place, which we call the top-down conservation narrative. And here we found that indeed scientific knowledge and formal rules may also support the conservation effectiveness for this biodiversity hotspot. In a follow-up study, and sorry, you don't need to look at this in detail, this is a knowledge network that we drew in a follow-up study on private farmers. We found out that many farmers didn't even know about the critical state of Renosterveld in South Africa. So in green here on the left side, you see individual farmers that had some patches of Renosterveld staying on or remaining on the land. And in fact, this habitat was only left on 5% of its original distribution in the Swartland, the study area that we looked at. Interestingly, there was almost no knowledge exchange between the farmers about adequate Renosterveld management or conservation management. And also there were very few links between government organizations and farmers. So these government organizations are highlighted here in light blue. So here were only indirect links through academic researchers that played an important role here in conveying knowledge. So most farmers here, and this is outlined in purple, would obtain their knowledge about Renosterfeld management from media or from active engagement of some academic researchers. Interestingly, we found a similar lack of information flow from official and conservation initiatives and government organizations to individual landholders in our case study on Coleus myrmidona in Romania too. So as Frank Wassen said yesterday, there is a great knowledge available that could directly be included in many conservation schemes. However, the integration of land use and conservation at the same time requires often a more nuanced understanding of interactions between species and their environment, and also a better understanding of the drivers and responses that influence the adaptive cycle of our systems. And at the same time, especially for rare species, we still lack out ecological knowledge on their development and their sensitivity towards management impacts, such as the impact of grazing patterns on the survival of larvae of Coleus myrmidona, or in a South African context, the impact of fire regimes on larval development. So a starting point can be to disentangle the components of our environment as social ecological systems, and to consider both biophysical and anthropogenic factors that can be spatially explicitly used for looking into butterfly distribution and abundances. And from such a perspective, butterflies can be seen both as interactions, but also as responses to a combination of these factors. And the system understanding may help to understand the dynamics of within this ecological subsystems, considering views on the entire life cycle of the butterflies, and include also views on movement patterns and therefore also the dispersal of species, and to center our attention towards these interactions between species occurrences, richness and composition to land management, structural complexity at a landscape scale, and dynamics over time. And social ecologically, my research has emphasized the importance of knowledge flows from decision makers at higher governance levels down to land managers. And here the butterfly can also act as boundary objects of the human-nature relationship, which may be an entry point towards an ethic of care and stewardship on which the state of the environment depends. And this may help to transform these interactions between humans and nature. But of course, governance challenges remain. As the research has shown, Renosterfeld in South Africa, for example, was conserved by law, but without control and enforcement. Many people also considered this biodiversity hotspot as wasteland because they couldn't see any value in it. So here, important work is to be done to develop a civic engagement towards conservation. Second, some targeted incentives such as agro-environmental schemes sometimes turn out to be relatively, limited for species that are outside the radar, as has been shown for the occurrence of Lycaena dispar on heterogeneous patches of agricultural land in Romania. Here, the farms are so small that they cannot actually apply for payments or incentives through agro-environmental schemes, so that our broad-scale conservation mechanisms to play out into agricultural use may have to be reconsidered both at an European, but most importantly at a national level. Then third, there are often scale mismatches that we have to deal with in conservation. For example, may biophysical boundaries not necessarily match the social boundaries that are related to specific administrations and fundings and other conservation instruments. And also, there is a gap to be closed between on-farm management, which may address the single actors, the farmers as conservation stewards, and across the landscape, which necessarily has to involve various actors. So these are the different mechanisms that may determine the effectiveness of conservation policies. Yeah, and to gain such a system understanding, we need to augment our interdisciplinary research modi and also to step into science-societal collaborations. And with this help, we may gain a system perspective that may spread from genetic diversity up to landscape heterogeneity and include also collaboration and knowledge management. So I hope I have convinced you that our effective, or to increase the conservation effectiveness, we need to rethink the way we approach our systems and to consider that people and nature conjointly form our environment. And this environment represents a social ecological system which consists of components that we need to study in depth, but also to connect these elements. And yeah, this way of thinking is probably a decision, as conservation is also a decision that we are, that we can take to bend the curve of biodiversity loss. With this, I'd like to thank you for your attention.
Yes, thank you very much, Jacqueline, for this interesting talk. Of course, it's a bit different than what we're used to when discussing the ecology of butterflies or butterfly trends and such. But it's, of course, a key element in trying to make conservation work by including, well, the perception of people of nature. So I think that's a very valuable contribution that you made here. We have some questions. And well, first, a very practical question is whether you talked about the intrinsic motivations being important for paying attention to conservation, but the main instruments in agri-environment schemes are, of course, often financial ones. And so, do you think, would it be sufficient to have just a financial incentive to work on conservation, or how do you see it could work best?
Yeah, so I think it's a bit, it's a tricky question. There is research out that shows incentives can first have a positive impact on farmers' action on the farm to support, for example, bird. The study is on bird diversity by Disnu et al. In 2013, and they have shown that it can work as long as the funding is there, but as soon as this financial mechanism breaks away, there is a detrimental effect. So as long as we can maintain this funding, then of course this is a very good tool to be used, but the sustainability of it is questioned. So at the same time, I think without any form of incentive, farmers cannot act the way that is always environmentally friendly, because there is so much competition on the market. So my my take on this is that there is not only one instrument that we should be working on right now. There are also campaigns to increase the awareness towards the insect decline, and there is a variety of tools that farmers can implement or that they may use. There are trainings and and financial support, and I think it's a combination of all of this. There is not one or the other.
Okay, yes, thank you. And well, question point that was raised during the discussions with Frank Vossen yesterday was that well, abandonment is of course a big issue in well, outside Northwestern Europe, and it's often driven by, well, individual farmers that stop their practices, but their land is often fragmented and private, kept in private ownership. And so perhaps you would need to move towards a different farming system of more cooperatives or so, where perhaps farmers lease their land to somebody else to continue the practices. So do you think that do you see that there are movements towards also different types of farming organization to deal with this question of abandonment? Of course, farmers have been adaptive for centuries, but I don't, do you have any view on their, let's say, their adaptive capacity in these times?
Well, in Germany, I've seen some examples where there were not really farming mechanisms maintained for farming, say, profits, but there were contracts that were specifically designed for conservation outcomes, and then small-scale farmers or farmers that would continue to use this land would gain some income through continuing to use the land. So there is more under a conservation agreement. So this is not really a farming instrument, it's really a conservation tool. And then in Romania, yes, we do see a few cooperatives emerging, but the question is if bringing together this land and then intensify the land use over a larger area, whether that's more beneficial than keeping some pockets of different stages of abandonment. But yeah, especially in the rural areas in Romania, abandonment seems to be a huge problem. And not only abandonment, but in some cases we even find that reforestation may also pose a threat. So there are also payments for carbon offsetting, which is then used for other income generation. And yes, that's a problem. And yeah, we need to keep an eye on the young people and they call themselves peasants movements. So yeah, I guess they would need all the support we can provide to maintain these structures.
Yes.
And to acknowledge the diversity and the contribution that they provide with, through this.
Okay.
And I would like to say that many farms in Romania still are so small that they are not eligible to apply for any further funding. So that's a bit of a perverse setting, which really poses limits to maintaining these really valuable farming schemes, farming landscapes.
Yeah, yeah. Yes, and there's also a question, well, from a Dutch perspective, here farmers still, well, often portray themselves as stewards of our open landscapes and of our green environment. And well, the problem that we see from butterfly conservation is that it's only green, nitrogen green, let's say, and that there's no room for more diversity. But there's of course a shifting baseline syndrome that's operating there, that, well, this has been a common picture from the 70s onwards. And well, if you don't know anything else, then then you have no reason to see that you're going in the wrong direction. So do you think how, or do you have an idea how to offer a remedy against that shifting baseline?
I don't know if it's a remedy, but I think, well, for first, we should probably pay more attention on the societal discourse and also not stigmatize the farmer as those people that are destroying a lot of things. Well, actually, they try to do a good job in providing food and to keep the land free of, you know, the weeds and stuff that we don't want to have because they may decrease the, or that's the thinking, decrease the yield. So to include the farmers and their values, maybe by also arguing in how far diversification strategies on the farm may also support, say, natural enemies for pests, or to prevent soil erosion, or to small steps to bring more color into the landscape. You talked about a green landscape, maybe in this close collaborations with the farmers, and also to suggest that a lot of small changes may also overall stepwise increase the diversity and restore many different pockets of habitat for various species.
Okay, thank you. I think we have to leave it at that. Thank you very much for your contribution. And we'll move on to the last presentation before the break. And that will be given by Thomas Merckx. And well, some of you probably know Thomas already from his work on the speckled wood butterfly in Belgium. But since then he has moved well across Europe, and also from butterflies to moths. And he is a sort of range shifter. And he has now moved to Finland. And so it was apparently very successful in overcoming habitat fragmentation himself. And he has moved on to work on urbanization effects on moths. And I think it's an interesting perspective from an area that's not so much urbanized in Finland, but maybe that gives a fresh perspective. And so Thomas, the screen is yours and your presentation is already up and running. So please go ahead.
Thank you, Michiel, for this kind introduction. And
welcome.
And
sorry, and the moderator will be Jurjen, who is coordinating our moth butterfly, our moth monitoring scheme. So yes, address your questions to him. Okay, Thomas, please.
Yeah, thank you, Michiel. So welcome to you all to this talk on urban Lepidoptera. And in it, I will focus on functional homogenization and local adaptation. But first, I want to show with these graphs that that urbanization is rapidly gaining force worldwide. As you can see here in this right graph showing global urban cover, you see that this, depending on the scenario, urban cover is forecasted to expand 2 to 6-fold by the end of the century. And here you see that the global urban population is set to increase from currently half to more than 2/3 already by mid-century. So urbanization is, it's not only limited to Finland here, but is indeed a worldwide challenge, as you can see from this picture. And if we look through the eyes or from the perspective of organisms living in our world, basically you can see urbanization as a real cocktail of several human-induced rapid environmental changes. So all types of pollution are going on, among which light pollution. There's also urbanization is also typified by very strong habitat loss and fragmentation, obviously. And then there is this famous urban heat island effect, typical of urban areas. So where you have these urban areas are typified by typically higher temperatures than their surroundings. So now, what does this actually mean for our butterflies and moths? And so here in this graph, this I've schematically represented 3 different Lepidoptera communities made up of different species. And these 3 landscapes are going to urbanize, to urbanized settings and to urban Lepidoptera communities. And so these areas here, these natural Lepidoptera communities, they have actually only two ways to go. So either they disappear, and this is because the habitat quality, quantity, and the connectivity of the habitat resources are becoming too low for their species-specific requirements. But some of them, they will cope with this urbanization process. And as we will see later on in the talk too, some of them may actually locally adapt to urbanization. And so our hypothesis here is that these urban species communities here, they are actually a non-random subset of these natural communities. And these urban ones, they actually share traits that have predisposed them to survive the city life. And this is what Hans van Dijk and I wrote up in this paper, where we tested for urbanization-driven homogenization, both taxonomically and functionally. And our prediction was that we would see declines in abundance, species richness, and species diversity, and that these declines would be driven by the loss of habitat specialist species, non-mobile species, and non-thermophilic species. And we did so by comparing both the butterflies and the moths. And so we came up with 2 additional predictions: that homogenization would be more pronounced at larger spatial scales for moths, because we believe that this is on average, that moths are on average more mobile than butterflies. And a third prediction was that homogenization would be more pronounced for moths, because this is a nocturnal, mainly nocturnal group, and so they would have this added negative influence of light pollution, of artificial light at night. And so we set out to test this in this study area here in the north of Belgium, which, as you may know, is a very urbanized part of the world, with 700 humans per square kilometer in this area here. And to get enough of sampling sites, we followed the 2-level subplot plot approach that you see depicted here. So in this graph, you see 81 very small local scale subplots set within 27 landscape scale plots. And at both these levels, subplot plots, we use the same 3 categories to get 3 classes of urbanization. So we have urban classes, semi-urban classes, and non-urban classes at these 2 levels. And then we moved away from this 2-level approach once we had our all our subplots selected. And to increase the precision with which we wanted to quantify the urbanization, we followed the multi-scale approach, which you can see here depicted for these 3 subplots. And so we quantified around the precise sampling sites urbanization, the built-up cover, at 7 different spatial scales, from a very local 50-meter radius all the way up to a 3.2 kilometer radius around each of these sampling sites. And then we sampled for butterflies and for moths. And for butterflies, all these subplots were sampled 3 times with the famous Pollard walk, giving us more than 4,000 individuals from 23 species. And macromoths weren't sampled in all these subplots, but in a subset here, and only in 12 subplots, but each of these sites were sampled 11 times. So we got like 3,000 individuals from 200 or so species. And here you see the effects of urbanization. So urbanization is depicted here. So this is the percentage built of cover. You see the top, you see strong declines in abundance, in species richness, and in diversity. And these numbers here to the right of the BUC, you see that they differ. They actually refer to the spatial scale at which urbanization has been quantified around the communities. So basically, each of these analyses were done 7 times with urbanization quantified at 7 different spatial scales. And so here is we only represented the models that gave the best model fit. And this differs between, depending on the trait, but also differs between moths and butterflies, as you can see. So here we got the best model fits at rather local scales, 200-meter radius, whereas for moths, this is typically at landscape scales. And these stars here, they represent the, they give an indication of the statistical significance. Moving on to functional community composition, we follow the community-weighted mean approach. So this is basically simply the mean of species-specific trait values of species in of all species in a community, and then weighted by species abundance. And so here for butterflies of larval host plant specialization is not significant, but for moths we see a strong decline in the community-weighted mean larval host plant specialization. We see increases both for butterflies and moths in adult habitat use generalism and in the trait for distribution, so that these urban communities here, they are made up of or typified by species that are more widespread on a Flemish scale than these rural communities, which also include more localized species. And moving on to graphs for mobility on top here, and wing length and wingspan for moths. Well, these traits here, they're actually proxies for mobility too. As you can see, you see strong increases with urbanization for all these panels. Again, butterflies, you see the best fits at local scale, for moths at a more landscape scale. And then finally, a graph on heat tolerance for butterflies. These traits, species-specific traits, were missing for moths, so we don't have this information. But for butterflies, you see that these urban communities are typically represented by species that are more heat tolerant than rural communities. And last graph here on the bottom is for habitat openness. So these communities, they contain on average species that are more linked to open grassland habitat biotopes than these rural communities, where you will also find a lot of woodland species, for instance. So what we conclude from this is that we do show indeed urbanization-driven biotic homogenization. We see these strong declines in abundance, richness, and diversity, and this seems to be driven by the loss of specialist species, non-mobile species, and non-thermophilic species. And we found very strong effects for butterflies, as you can see here. So if we would, for instance, compare sites with a 25% build-up cover in the surroundings, which are highly urbanized sites, comparing them to sites without any urbanization in the surroundings, then we see like 85% decline in abundance, 60% decline in richness, 25% decline in diversity. We see that the average mobility of the community goes up by a quarter almost, and the wing length, for wing length by 7%. But all these values are stronger in moths, as you can see. So it's even stronger, these effects of urbanization for the moth taxon. And this we think may be due to these added effects that this nocturnal group is experiencing of artificial light at night. And then also all these, for butterflies, the best model fits were always at a local scale. For moths, it was typically at larger scales, and this is linked to the on average higher mobility of this group. So now focusing only on moths and on body size. So here we have wing length as a proxy for body size. So you see that these communities here, urban communities, they consist on average of species that have a 20% larger wingspan than these rural communities. And in this paper, we included both these community level effects, but also looked at species level effects on body size. And so for 35 species in total, we had enough data to look into this. And we found that none of the species actually showed a significant decrease in body size with urbanization, but 12 species, so a third more or less, showed an increase in body size with urbanization, and they are all depicted here. And what you should also notice here is that the best model fits were from the very local scale, 50-meter radius, all the way up to the larger spatial scale and everything in between. So there's a large variation there. On average, for all these 12 species combined, for every 5% point increase in urbanization, these species got like quarter heavier body mass. Okay, so what we conclude from this is that the size filter that urbanization is actually works at two levels, so both at the community level and intra-specifically, so that smaller species are lost from communities, but also in some species, we are losing smaller individuals. And because Lepidoptera are a key player in all terrestrial ecosystems, and because body size is central to many species interactions, we believe that these urbanization-driven size increases may actually start to impact the functioning of urban ecosystems. We're thinking here about diurnal pollination, nocturnal pollination, and food web dynamics, because moths and butterflies, they're really important players in ecosystems, as we know. And have stressed a lot that that we saw effects at different spatial scales for different groups. So if we're thinking about mitigation of these effects on body size, we need to act at multiple spatial scales indeed. So we can act by providing urban green infrastructure. We need to provide lots of them. And of course, it needs to be of high quality. So you need to provide a high diversity and high abundance of native vegetation for this group, and importantly, implement it at multiple spatial scales, so from the local street level scale all the way up to the city level scale. And then you can combat some of these effects that we are seeing on body size, both at community level and within species. So in this final part of my talk, I will talk about adaptation. I've already alluded to it in the introduction. So we saw that smaller individuals are getting lost within some species. And you have to know that within butterflies, but also in moths, there is a positive link between body size and mobilities. So larger butterfly species, they are typically more mobile species than the smaller ones. And the same applies for individuals too. And so this indicates, so that we're losing these smaller individuals, this indicates that urbanization is driving shifts towards larger and hence more mobile individuals in order to combat this low connectivity that is typical of ecological resources in urban settings. So here you see a very dispersed configuration of these green habitat resources, and you can imagine that it's only the mobile, the large species and individuals that can actually deal with such a setting. And this actually goes against the general trend for most taxa, as you can see here, where urban communities typically start to consist of smaller species. And this is because of the urban heat island effect driving these shifts because of metabolic reasons. But because there's this positive size mobility link in Lepidoptera, here for butterflies and for moths, we see actually the opposite trend. So believe that there is actually scope for genetic differentiation too in body size and hence in mobility between rural and urban Lepidoptera populations, but this is material for future research. Urbanization now has recently emerged indeed as an agent of evolutionary change. Actually, it's no surprise because urbanization can have huge impacts on fitness. And there are a few examples of adaptive evolution in a few plant species and also some animal species. And here are the key examples for Lepidoptera. So there's of course this classic example of peppered moths evolving darker moths in response to soot pollution, which is also linked to urbanization. This is a very classic example. But then there's also the work by Florian Altermatt, who showed that these, when he took larvae from light polluted areas of these small ermine moths and from completely dark rural areas, and when he released these to light, he noticed that in both sexes, the individuals from the light polluted areas, they were flying less to light than the individuals from non-polluted areas. And so you can imagine, this flight to light behavior and also camouflage has obvious fitness consequences via predation of bats and birds, so driving selection. But I want you to know that phenology, and so the seasonal plasticity that underlies this, is also critical for organisms, even more critical maybe. So actually, if you imagine a larvae mid-season of a multivoltine species, and it has to decide whether to either develop directly or to enter diapause, it's actually a decision on life or death. And this decision is skewed by day length and by temperature. And we know from earlier work that these photoperiodic reaction norms that driving this decision, they do respond to climatic change. So Altermatt has shown this for lots of European butterflies and moths. Here you see 4 example species of this. If you would compare these flight curves before 1980, you see the dashed lines versus the solid lines is a situation after 80, you see strong increases in volatilism. So there's a higher investment in this second generation or in later generations for lots of multivoltine butterflies and moths. But the potential for photoperiodic reaction norms like this to evolve in response to urbanization instead of climatic change has not yet been tested. And that's what I recently am involved with. So this graph here shows you like a typical growing season from spring to winter. On top, you see a natural environment, and you see how day length actually evolves in a way over the growing season. You see an individual entering here as a pupa, and it closes as an adult. It gives rise to next generation. And these dots here, they represent the pupation. And if pupation happens before this dashed line here, before the critical day length, so that's the day length at which 50% of a population would go into diapause. If this pupation happens before, then the individual will decide to go for direct development, and it gives an extra generation, some of which will make it to winter in time in their diapause stage, others won't make it. And if this pupation stage is reached after the critical day length, then they go for diapause to the next to winter. Now, under an urban warming scenario, you see that you get more, there's scope for more offspring. And this happens because the flight season starts earlier due to this urban heat island effect. The development is also speeded up, so it's shorter here than under normal conditions. And the growing season is also pushed backwards because of these increased temperatures. And you see that also these late individuals, they now safely make it to winter time. Now, if we would consider a light pollution scenario, then the perceived day length may be longer. for our Lepidoptera. And so this actually means that this critical day length is pushed to the right, is pushed later in the season. And this can have huge effects on butterflies. You see that here now also these late individuals, they're also tempted to go for a direct development, but they won't make it in time. So this is a typical example of what we would call a developmental trap. And so you can see from these hypothetical scenarios that there's a lot of scope for selection now to kick in. So if selection would pull back these critical day lengths to the left, to longer photo periods earlier in the season, it would compensate for this developmental trap. And for the urban heat island scenario, actually, there's also scope for selection, but it goes in the opposite direction, as you can see here. So there is scope to push this later in the season, the critical day length. There is still scope to get direct development and to get them safely in time to this winter diapause stage. And so this is what we started to test last year. So here you see a schematic representation of these two opposing evolutionary patterns. So the urban heat island should select for direct development to continue later in the year. So that's adds shorter photo periods, because this increases the fitness of genotypes that are making use of this extra time. Whereas Allen light pollution should lead to this compensatory selection for earlier induction of diapause, so at longer photo periods. And we tested this with two Lepidoptera, one moth species, the lattice teeth, and a very common butterfly, the green-veined white. And we got females from the Stockholm city region and from Helsinki city region, which you can see here, these dotted dots, these colored dots there in the map. And we bred them in the lab to the F2 generation for the lattice teeth and to F1 generation for the butterfly. And we got them in climate rooms, all at the same temperature, but at 5 different photo periods. And we simply compared the probability of direct development among these different groups. And what we saw is really indicating that there is selection driven by the urban heat island effect and not by artificial light at night. So for Clatrata, for instance, you see a huge effect of in Helsinki pushing this critical day length 1 hour to shorter photo periods. And this actually corresponds in the field with a 9-day later critical day length in Helsinki. For Napi, these shifts are smaller, but still substantial and biologically highly relevant, and they correspond to a 5-day difference, more or less, in the field in Stockholm and in Helsinki. And this is actually the first evidence, we think, for genetic shifts in such photoperiodic reaction norms following urbanization. And this is very recent material, so it's now under review. So I'm asking that I shouldn't put this on social media. In addition to this, we also so we got these nice results, and then we said, okay, let's now see in the 3 biggest cities in Sweden and the 3 biggest ones in Finland. Let's see if we can pick up something like that from in situ flight curves. We focused on the same 2 species, and now we used long-term citizen science observation data, and also data from standardized monitoring for these moths and this butterfly. And we started comparing flight curves from urban versus rural populations. And this is what we saw. So the flight periods for both species, they were typically longer, much longer in urban zones and in the rural ones. and also they end later in the season in both species. So for instance, for Helsinki and Stockholm, the last flight day was 5 days later, more or less, than in the rural surroundings. And this actually corresponds very well with the effect what we found experimentally. So these flight curves, they are suggestive of partial extra generation, and they may suggest that similar genetic shifts have occurred elsewhere in other cities. And so finally, so we expect that this urban warming may drive genetic shifts in critical day lengths and hence increase investment into late generations and volatinism in at least in multivoltine species in Lepidoptera globally. And I'd like to finish with thanking all my co-authors and all the citizen scientists involved too, because their data is very important for this kind of research. And especially I want to thank you for your attention to this talk.
Yeah, thank you very much, Thomas, for a very interesting talk, and also, well, really new developments that remain to be investigated. So I think we're really entering a new field here, and well, and apparently you do find cities in Finland even to investigate. So there were a few questions, and one of them is whether the difference that you found in the scale of effects between butterflies and moths in your Belgian setting could also have been a sort of artifact from difference in sampling intensity, because of course, well, I guess with moths you have a larger species pool, and then you might have higher requirements to detect effects that you noticed earlier in butterflies. So it could be a sort of a sampling effect. What do you think about that?
Yes, it's a good point, but I wouldn't agree with it, because so as you noticed, so this moth sampling happened with light traps, and you may think that they are attracting moths from far away because you can see these bright lights from far away. But yeah, I've shown, and other people have also shown, that this attraction radius of these light traps is actually quite local scale. So a few tens of meters on average. From within such a radius do they actually start to attract moths that are passing by. So actually these samples, these moth samples, they actually represent the very local habitat. So yeah, unless I understood the question a bit differently, I don't think that there's any bias there by the way we actually sampled. Actually, it would be a bit the opposite, because for moth sampling as a passive method, it's really at one spot that the sampling is happening, whereas for butterflies, we followed these pollard walks, so we walked around for 20 minutes at low pace. So actually there, you sample a slightly larger area than for moths. So I really think that these different scales of effects that we're finding between these two groups are actually real. It's also been shown in other research where you find, like, if you look at the effects of agricultural intensification of moths in general, there too you find effects at a 800-meter radius scale, so like a 1 kilometer radius scale. You see the biggest effects happening, and this is a bit smaller for butterflies too.
Okay, yeah, thank you. Yeah, I think that that does answer the question. And I have another one that I will read, because it's quite a complicated question on the beta diversity. Oops, yeah. So whether you find a sort of whether your species found in urban areas are really a subset of those from the rural areas, or if you find a pattern where differences in between species of urban and rural areas actually are, well, from a different species composition. So then, of course, that also has consequences for local adaptation. So what about these patterns?
Yes, that's a nice question. So typically, you haven't really looked at these effects of beta diversity, but from just knowing the species there, it's pretty clear that these urban communities are a subset of the whole community that is out there in the surrounding landscape. So you might have one or two specialist species that are specializing that you will find only in cities and not in the surrounding landscape. But most of the case, it's really like it seems that this filtering effects has happened, and that you get a small subset of from the regional community. Of course, there are a few like exotic species that might, or species coming from the south that will first establish in the warmer cities and then spread out again. But these are kind of exceptions, and so yeah, it's really, as I said, like a subset. Yeah, these urban communities.
Okay, thank you. I think we have to leave it at this. And we'll thank you very much, Thomas.
Thank you.
And we'll move on to the coffee break. So please take your coffee and visit one of the breakout rooms, and then you can have a chat with others. And we'll continue with the presentation by Vlad Dinka at 11:30. So enjoy your coffee and your chat. Bye-bye. Okay, I think we're ready for the last two sessions of this symposium. I hope that everybody enjoyed their coffee and breakout room. And we'll continue with a look at the diversity side of butterflies. And then from a genetic perspective, and Vlad Dinka will tell us all about it. Vlad is working at the University of Oulu and has a position as academic research fellow in evolutionary biology. And he also, like Thomas, he completed a successful northwards expansion. I don't know whether it was related to climate change, but Vlad has roots in Romania and then moved around in the Mediterranean environment, but now has landed in a boreal environment. We'll see if this also gives a different perspective on things. So, Vlad, I would like to ask you to share your screen and start your presentation. Please go ahead.
I am being told that the host disabled participant screen sharing.
Oh, that's not a nice thing, but I'm sure that Kars will remedy that.
In the meanwhile, just to say thank you for the introduction, Michiel. So, well, you know that I have done my PhD in Spain, in Barcelona, so I have been in the Mediterranean. So now I thought it's also nice to try the northern part of Europe, no? So perhaps I can better understand the dynamics of butterflies, you know, and of their distribution. So both are very, very interesting.
Yes, yes, I would agree.
Yes, now it works.
Yes, there you go. Okay, great.
Okay. So I understand you can see and hear me properly.
Yes, you just have to go,
Yeah.
yes, in the presentation mode. Okay.
Let me just choose the pointer. Okay. So hello, everybody. I'm very happy to be here today to participate to this very interesting meeting and to have the opportunity to talk a bit to you about some of our research on European butterfly diversity and conservation using genetics. So before going further, I would like to mention that the results that I present here are the outcome of an excellent collaboration between various colleagues. You will see their names as the presentation progresses, but I wanted here in the beginning to thank them for that. So during the last approximately 15 years, we have been investigating or exploring European butterfly diversity using DNA barcodes, so mitochondrial DNA. And we started at a more regional scale with Romania in 2011, then with Iberia in 2015. And just a month ago, exactly a month ago, we managed to publish the first comprehensive DNA barcode library for the European butterflies. So you can see here the sampling that we had for this study. The orange dots represent sampling sites. Of course, for most of them, there is more than one specimen or more than one species that are sampled. In total, we gathered over 22,000 DNA barcodes for 459 species, with an average of 48 specimens per species. So this represents 97% of the European butterfly fauna, not counting the species endemic to the Macaronesian Islands. So these have not been included in this study. So of course we lacked a few species. I said we cover 97%, but most of them are really with marginal occurrence in Europe. So I would say they are not really representative for the European fauna. Most just occur at the Urals in Europe. So this information, I should also mention that, of course, was obtained by accumulating previously published data, also from other colleagues who published some regional DNA barcode libraries, and adding about 5,000 new DNA barcodes. So we got this, I would say, quite massive data set for the European continent. And of course, having this data, we were very curious to address a few general questions, so to assess a few general patterns. And one of the first things we asked ourselves was, with this kind of sampling of over 22,000 sequences, how much of the genetic diversity of European butterflies has been captured by this data? And doing the math, we estimated that we captured about 62% of this diversity. Why only 62% and not more? That is because when we looked at the frequency of haplotypes across European butterflies, we found that most species have a few very common haplotypes, so dominant haplotypes that are found in many specimens, as you can see here, and many rare ones. So this means that if you wish to capture a high percentage of the genetic diversity in European butterflies, you probably need to sample a lot of specimens because many of these haplotypes appear to be rare. So the likelihood to capture them with a small sampling, it's low. And this is, I think, an important message also for, in general, for studies that use mitochondrial DNA to study European butterflies. It requires an extensive sampling and a good geographic coverage to increase the likelihood of having a good representation of genetic diversity, so as high as possible. I should also mention that although there are these many rare haplotypes and so on, they usually involve single base mutations. So we are talking in general about little genetic divergence. Another continental scale pattern we wanted to assess was the relationship between latitude and genetic diversity in European butterflies. I mean, intraspecific genetic diversity. And as you can see here, this is a graph showing genetic diversity across latitudes. So you can see that it is highest in the southern parts of Europe, here on the graph, and it gradually decreases towards the north. Now, I think we already know from, especially from case studies, that this trend was to be expected. Why? Because this is likely an effect of the glaciations, when, of course, during the last glacial maxima, the butterfly fauna retreated south in the so-called refugia, mainly the Iberian Peninsula, the Italian Peninsula, and the Balkans. And then during the warmer interglacials, most species will start recolonizing the north. And of course, just a subset of these reservoirs in the south will migrate north. So of course, they will carry with them just a portion of the genetic diversity of the species. Hence, you get this gradual decrease. I think the key message here is that this graph shows and establishes the generality of this key biogeographic pattern for an entire taxonomic group for the whole continent. So this is a clear trend for the entire butterfly fauna. So I think this is the power of this analysis here. So we can be quite sure that at least in terms of mitochondrial DNA, there is this decrease in genetic diversity from south to north. So I showed a few general patterns, and to say it briefly, we now have an unprecedented overview of the mitochondrial DNA diversity for European butterflies. And we can literally start mapping genetic diversity for individual species. Here I'm showing just an example for the butterfly Lasiomata mera, where we use the RGB color space to map genetic structure. And it's very visual, I think, so it really helps to quickly interpret the distribution of genetic diversity, because we see three main lineages. One with green in mostly related to Iberia and a bit to southern France, one with yellow in the Italian peninsula mostly, and with red, a more widespread one from the Balkan, that interestingly, apparently with the current sampling, was the only one that also colonized Northern Europe. Of course, this is just a case, no? It's Lasiomata mera. There are several other patterns that we see depending on species. Of course, other species show much less structure. So it varies on a case-by-case basis. But I think this kind of information, it's already very valuable also for conservation, because we can start to understand at a large scale what is the distribution of these genetic patterns, and this kind of information, I think, can be integrated into conservation programs, for example. And now I would like to briefly talk about an example that, in my opinion, illustrates quite well how genetic data can be used to help conservation programs. And it is about this butterfly Melanargia russiae. And its story is that it has become extinct in Hungary over 100 years ago, more precisely in 1913. You can see here the last known localities in Hungary. And there was a plan by the Hungarian nature conservation authorities to try and reintroduce the species to some of the sites where it formerly occurred in Hungary. And of course, one of the key aspects when you're thinking about reintroduction is habitat quality of the target sites. And as far as I understood, this was deemed to be quite good. So there was potential for successful reintroduction from this point of view. However, another aspect that has been considered as important was to, if possible, to obtain some information on the genetic features of these extinct populations, because ideally what one would like is to use as source populations some that are genetically as similar as possible to the extinct ones, because in theory this would increase their chances of survival in their new home. So for this purpose, we joined forces with Joel Balint and other colleagues Jorbarin, working at the Hungarian National History Museum in Budapest. And we tried to DNA barcode some of these specimens from the extinct Hungarian population. So over 100 years old specimens. You can see one here to the left. And here you can see some images of the places in Hungary where the reintroduction was planned. And we managed to obtain some mitochondrial genetic data from several specimens. And of course, the key was to compare these to extant populations across the range of the species. So to see which are more similar to the Hungarian ones. And we mapped genetic diversity in a similar way as done for Lasiomata mera that I exemplified before. And by the way, this kind of genetic maps were done by my colleague and close collaborator, Leonardo da Porto from the University of Florence, Italy, and with whom I often work together. And you can already see that Melanarchaea russea across Eurasia has three main lineages. One with green, again, restricted to Iberia and southern France. One with blue, interestingly, apparently endemic to Sicily. And a much more widespread one with red here that ranges from Italy up to eastern Kazakhstan. And we can see the red dot here for Hungary. So we can already know that if we want to reintroduce the species in Hungary, we should probably best avoid Spanish populations or southern France or Sicilian ones. So we are already narrowing things down a bit. However, we wanted to take things, and by the way, this Hungarian population displayed a possibly endemic CO1 haplotype. It is true that this haplotype displayed only one mutation to the nearest one, so it is not highly diverged, but still, if this was indeed endemic and it has been, the species, it has been extinct from there, we have forever lost a small fraction of the genetic diversity of this species. And this is, of course, not good. Imagine if this repeats in several sites, this would lead to genetic erosion, which is certainly not a good thing for the chances of survival and adaptability of the species. So coming back to the next step, we also did the climatic similarity analysis. So basically, we wanted to see where across the range of Melanarchaea russea, where there are extant populations that inhabit a climate as similar as possible to that in Hungary. And you can see here, of course, the more similar the color to the red that occurs in Hungary, the more similar the climate in the other parts. So basically, the populations from eastern Ukraine and neighboring Russia inhabit areas with a climate very similar to that in Hungary, where the target sites for introduction are located. So basically, by combining the genetic data above with the climatic data, it seems that the most reasonable choice of source population would be populations, as I said, from eastern Ukraine or neighboring Russia. So I think this quite clearly exemplifies how using genetic data, museum material, again, very important in this case, and other analysis, in this case, climatic analysis, can support informed reintroductions that should maximize the chances of success. Okay, so I gave these examples and I showed a few general patterns. And we have seen also here at the conference, and we know that unfortunately biodiversity is still declining. So we should not only study, biodiversity for the sake of research, but we should actually accelerate this process also because we need to be very efficient in better preserving it. And I listed here just a few of the ways we could we could use genetics to help. And one would be to identify the so-called evolutionary significant lineages, or ESUs. These can be at intraspecific level, but they can act as units for conservation. And one of the most basic things to do would be to better document their distribution. And then, of course, from there, we can go for more refined approaches. Also, we should better understand what is the fraction of cryptic diversity. And once we know it, what fraction of this diversity may require protection. So we know that cryptic species are present in the European butterfly fauna, and probably a few others await discovery. And I just listed here to the right some examples that were recently, quite recently published using a combination of molecular and other sources of data. Many of them started from mitochondrial DNA, which gave the first hints, and then they were investigated in further detail. And from this very recent study with the DNA barcode library of the European butterflies, we can still see that we have 33 species that display more than 3% maximum intraspecific divergence, or 56 if we lower this to 2.5%. So this is still quite a number. Of course, we will wonder ourselves how much cryptic diversity still hides in there. Certainly not all these species will represent cryptic diversity. Of course, we will need to do detailed studies on that, but some of them I suspect yes. How many we will have to see with further research, but I think this already gives us some clues, so it can help us direct our research in the needed directions. And then on the other, at the opposite end, we also noticed a very noticeable a very notable percentage of taxa that are currently accepted as species in Europe that lack interspecific divergence in mitochondrial DNA. More precisely, 15% of the European butterfly species are sharing barcodes. It is a high percentage. And of course, what does it mean? It could be due to biological processes, so the inability of mitochondrial DNA to distinguish these species, it can happen, and we know in some cases it is certainly the case. But we estimated that about 2/3 of these species may actually involve, let's say, insufficiently resolved taxonomy. So they are taxa that are not very well differentiated and that are regarded as species based on a rather weak evidence. So I think this would need further investigation to really establish if we can actually refer to them as species or not. So I think 2/3 shows that there is still quite some taxonomic uncertainty in European butterflies, as much as we love and study them. And of course, if we are dealing with this kind of significant taxonomic oversplitting, this can also lead to a notable conservation load. So I think it is good to clarify things as much as possible in order to prioritize conservation actions. Okay, so until now, I have talked about DNA barcodes and mitochondrial DNA because this is the kind of data that we have at continental scale, and it's something that we use for general patterns. But this is belonging to the so-called Sanger sequencing. Sanger sequencing is basically generating one sequence per run, but it is the older technology. In the last years and evolving very rapidly, we have the so-called high throughput sequencing. And there are various methods included here, but it's called high throughput sequencing, of course, because it generates massive amounts of genetic data. And I made here a table that is very, very brief, very simple, and this tries to show some of the key differences between the two technologies. To sum up, Sanger sequencing involves relatively lower lab startup costs. It is generally more permissive depending on the marker targeted regarding DNA quality. It has a lower sequencing rate, at least for the moment, but as I said, it has lower computational requirements because, of course, we are dealing with much less genetic data, but it delivers a low amount of genetic data, one sequence per run. However, by contrast, high throughput sequencing requires more initial efforts to set up the lab. There are much higher expenses. It is usually very computationally demanding because we get a lot of genetic data, and usually many of the analyses have to be run on clusters and they can take several days for some kind of analysis. And it is generally more demanding in terms of DNA quality. So it tends to be trickier with older samples, but I'm saying it's not in absolute terms. So there can be obtained high amounts of genetic data also with older samples, but these are just, let's say, trends. And also high throughput sequencing technology is evolving very rapidly. So things are improving quickly and the prices are also getting lower. So it has clearly a huge potential for the future. And one of these methods of high throughput sequencing is the so-called double digest restriction site associated DNA sequencing, or briefly ddRAD-seq. So let's stick to ddRAD-seq. As the name implies, it involves the digestion of DNA in 2 steps with the restriction enzymes, and it generates random data across the genome. Importantly, it doesn't need a reference genome, so it can be used with non-model organisms that don't already have a reference genome, a high-quality reference genome. So this is an advantage in many instances. It can provide quite high resolution because it can generate easily thousands of loci randomly distributed across the genome, and it's very good for intraspecific level or closely related taxa. So not for deep-level phylogenies, for example. And I want just to quickly go through an example using this kind of sequencing approach. And our hero here is the butterfly Melitea didima. We knew already since 2015 that this species displays a very notable level of intraspecific mitochondrial DNA divergence. And in 2015, we found 4 main lineages, but in 2016, Mazenkova and Luktanov found 11 CO1 lineages, and they found that some of them display up to 7.4% minimum distance, genetic distance in mitochondrial DNA, which is really remarkable. It is one of the most remarkable cases of intraspecific mitochondrial diversity in European butterflies, if not the most, let's say, striking. They also found that it displays low variability in the chromosome number, although the samples were limited for that. So we really thought this is an interesting case to investigate with this kind of genomic methods and see if these mitochondrial patterns are actually reflected in the nuclear genome as well. And if we maybe deal with an extreme case of cryptic diversity. And we had 93 specimens for which we had, of course, CO1 data, so DNA barcodes, and this ddRAD-seq data. And quickly, the geographic distribution of the mitochondrial lineages. In our sampling of 93 samples, we had 10 lineages. 2 were different from the other paper of Mazenkova and Luktanov. And so this means there are in total 13 lineages in this species, really remarkable. And just without going into too many details, because the distribution of these lineages is very complex here, some are in allopatry. So I used different shapes for the different lineages. Like North Africa here, you can see Sicily, endemic. And several others were found in sympatry or near sympatry. You can see, for example, here in Italy, in southern France, in Spain, in the Balkans. So it was a very complex distribution with several cases of sympatry. Okay, so what happens when we looked at the nuclear genome using this ddRAD-seq? Well, you can see here the same symbols, the same shapes used for mitochondria, so they represent the mitochondrial lineages to compare, and the colors represent the lineages, the clusters that we got using ddRAD-seq. So basically, we got 5 clusters with ddRAD-seq. Only 2 of them perfectly matched the mitochondrial data, which is North Africa, here with orange, and Sicily, here with violet. And the other 3, you see, encompassed multiple mitochondrial lineages, but we didn't find any case of sympatry in the nuclear. You can see there is a geographic clustering here. No, we have Iberia, we have this part with Italy, south of France, a bit of the Alps, and then we have an eastern lineage in the nuclear, but nothing in sympatry, unfortunately, although, of course, when we designed the sampling, we included sympatry based on the mitochondrial data because this is what we knew at that time. Please also note the number of loci here. This is over 22,000, and we had almost 3.5 million base pairs compared to this to the 658 base pairs that you get with barcoding. So the difference in the genetic data it's massive. Of course, here it's true that we have a lot of missing data because it depends on how you filter it. But even if we would be stricter, we will still get several thousands of loci. So still many times more information. So basically, what we found here is this notable genetic differentiation within Didyma. Especially the Sicilian populations appear very well differentiated. And honestly, although we need further study, I think they could represent a cryptic species endemic to Sicily. And that already might raise questions about the conservation status of these populations. But overall, we need further analysis, especially focused on the contact zones, because of course, I believe that it is possible that at least on the mainland in Europe, these lineages, the brown and the blue and the black one meet. And it is key to find such populations and assess levels of gene flow and levels of admixture to really see if we actually deal with cryptic species or not. Okay, so, and I think also this case shows not only the resolution and the resolving power of high throughput sequencing, but it also shows that even with such wealth of data, in some challenging circumstances like allopatry, some fundamental questions and problems remain. I mean, genomics will not solve every question just because we have more data. Of course, it gives us much more confidence in the patterns because we have a lot of data. But some conceptual problems still remain, such as delimiting species in allopatry. Okay, so for the future, we are already starting to investigate some types of EU conservation concern, such as some of those listed in the Habitats Directive, namely Agriades achilo, Erebia polaris, and Poliomatus eros, regarded nowadays as species. But you can see, for example, that even in the Habitats Directive, 2 of them were listed as subspecies, and Eroides, according to the current taxonomy in Europe, is not even recognized as a species at all. So basically, we are having a species on the Habitat Directive that is not recognized by the taxonomists now. So I think it is important to better understand the evolutionary history of these species, and of course, use this genetic data to improve their conservation and to actually even decide if they are still worth protecting, of course, with priority at EU level. It doesn't mean they cannot still be valuable for conservation. Furthermore, we see that they are either share DNA barcodes or display very low levels of interspecific divergence. So we are very curious. We are looking forward to getting results on such cases. And to finalize, I would say that what I noticed, and we also discussed this a bit in the breakout rooms yesterday with a few colleagues, is that there is still a gap in European butterflies between conservation efforts and genetic data. And I think there is a great potential to combine and to strengthen ties here and to use genetic data more into helping conservation programs. And perhaps it would be a good idea even to create a working group of conservationists and geneticists trying to, first of all, to make it clear for us which would be the best ways to use genetic data to help conservation, and then as a second step, to see what can be done in practical terms. And with this, I would like to, of course, thank the funding sources for my research, which is the European Commission through the Marie Curie program, and now the Academy of Finland, which is currently supporting my research. And of course, I would like to thank very much to all the colleagues who supported our projects. And of course, I thank you very much for your attention, and I am happy to take questions.
Okay, thank you very much, Vlad, for a very interesting talk from a different perspective, but with clear relevance for conservation. And I agree that it would be perfect to work towards combining the information on conservation status and genetic diversity. And one of the questions that was raised was then, because it's perhaps not getting easier, whether you should focus on sort of evolutionary significant units, so focus conservation on more the units, or rather focus on the promoting the mixing of different populations in order to, well, sort of preserve the power for adaptation. Do you have views on that?
I see. Interesting question. Yes. Well, of course, it also depends it will be depending on a case-by-case basis, no? So on the patterns that each species displays. But I think in general, we should, of course, aim to preserve as much of the genetic diversity as possible. So I think we will be able soon to find hotspots of genetic diversity at a more regional scale. Of course, the more data you gather, the more at the final scale you can go. And then clearly, I would say it is indeed desirable to, let's say, if you establish a protected area, to choose, if possible, areas that encompass as much of the genetic diversity of that species or of those species as possible. So clearly, going for preserving diversity in general is the way. And I don't know if I also understood the question regarding to these evolutionarily significant units. Because, you know, there is another question here. Do we want to focus on species or on this kind of evolutionarily significant units, which are, well, not necessarily species. I mean, they are interspecific. So this is another question, because we know that mostly even legislation and in general, also the general public, I think perceives easier the concept of species. I mean, you know, it's also the basic unit also for conservation, I would say, at least for the moment. And I think that both have merits. I mean, in the end, to preserve evolutionary significant units, I think first we need to really know them well. And we are starting to see this. But you know, we have mitochondrial data, but in terms of nuclear DNA, and especially using this kind of more recent techniques, we still have a lot of work to do. I mean, we are starting to get data, of course, it's proceeding, but it is a matter of also time. And I think it's good first, before going too much into this, to really understand well how they are, you know, where they are and what is their, you know, relationship with others. And it can be done. Technologically, it's possible. It's a matter of resources in the end, of financial resources. I mean, in theory, if we would have unlimited financial, let's say, resources, then you would have a lot of manpower and you could do it.
Yeah.
So,
well, the potential is clear anyway. And well, there are more questions, of course, but perhaps in the breakout rooms there will be time to discuss them further, because we need to continue with the final part of the program before the breakout sessions, and that is given by Chris Van Zwaay, my colleague and roommate at Butterfly Conservation, as far as we can work in the same room in these times, of course. But of course, well, you all know Chris from his long-standing work on butterfly monitoring and conservation. And Chris is increasingly working at a European scale of monitoring in the ABLE project. And I should not forget to note right away that Cristina Sevilleja is doing the moderation, and that is also a special pleasure to mention her, because she has been at the heart of the ABLE project, making contacts everywhere and giving workshops and involving people. So that's a very important person in the background. Okay, so Chris, please give your outlook for the future of butterflies in Europe.
Okay, thank you. So I'm gonna try to summarize some of your remarks, some of the talks, and also have a little thoughts about how things will change in the near future. First, some nice slides. This is also to get you in a good mood, but also to show the huge diversity we have in Europe. So from the high Alps over the semi-deserts in the south of Spain, but also these beautiful wetlands in the Netherlands, where we have a large couple, Icena dispar, flying together with Purple Emperor, Apatura iris in the woodlands to the left, and the beautiful woodlands in this case in Bavaria in Germany. Why do I show this? Because this is of course also Europe, especially in the Atlantic zone. Many of the former beautiful areas have been changed into this type of landscape. And we've all talked about that before, but we have to come back to some of these points again. Because when we look at the future of butterflies in Europe, we have to think of questions like, how are butterflies doing? Will we still have butterflies in 20 years? And how can I help butterflies? And of course, we have some instruments to think about this and to work on this. Red lists is one of them. I'll show some examples later, and there is a good chance that the red list will be renewed in the coming few years for butterflies, and there will be a red list for moths. Of course, we have the monitoring, and there is a lot of research on butterfly ecology going on. But before we answer the question, how are butterflies doing, it's always good to think of compared to when and compared to what, because that really matters a lot. This is a photo which should be about a primeval forest in the east of Poland. Of course, when you go back long enough, most of Europe actually consisted of this type of woodland, but afterwards, these nice old plates from schools, large parts of Europe were colonized by people. And of course, we also had the Middle Ages. So what is the period we are looking at? This is a famous picture by Koekoek, a painter of the Netherlands of the 19th century. Interestingly enough, he lived in the area around Wageningen, well, a bit further east, but and he made these beautiful paintings, but we all wonder where he saw these trees, because if you look at maps of these days, there were almost no trees to be found. So what was real? What is what we are comparing the landscape to? And what we do know is that, of course, not that long ago, this is really a photo from the Netherlands from more than 100 years ago, but it also shows that people were very poor in those days, and probably in the fields, I make this, yeah, in the fields behind, it might have been really swirling with butterflies. But for the people who lived there, they had other worries. So how are butterflies doing? Let's look at the two main instruments we have for that. And then later we can try to extend is what we can learn from the past and how we think that this will extend to the future. So we have the red lists. We have the last red list is of 10 years ago. It's good to realize that a red list focuses on the risk of extinction. A species can decline without making the getting considered threatened. Because you really need a big decline of more than 30% in the last 10 years, which is really a lot to get into one of the threatened categories. Of course, there are other ways to categorize as threatened. With monitoring, we can look back a few decades and we can try to learn from that to look into the future. First, the red list. It was made, this is the 2010 red list, and actually it showed a rather limited number of butterflies which are considered threatened. If you do it in a pie like this, you can really see that just about 1/8 of the butterflies would fall into a threatened category. So all is well with butterflies? No, all is not well with butterflies. Some we know quite a lot, but for many we don't. I'll show some examples. This is Corias myrmidona, a typical butterfly mentioned on the Habitat Directive annexes, and also of what we know that it has been in decline for a long time already. A lot of conservation work has been done. So Jacqueline Loos, who talked earlier this morning, she was also very active in this restoration project in Romania on that species. We see also Matthias Dorlek. And they produced a nice leaflet how to preserve the last Coleus myrmidona in Romania. So from that species we know a lot, but for other species we also know that knowledge has changed. So Erebia christi on the last red list was considered vulnerable. This is a relatively rare species, actually only found in a small part of the Alps. And work has been done on that species recently. And maybe it's hard to see the Matera 2 people hanging on ropes, climbing these almost vertical rocks in the Central Alps. And they did a lot of research on the Erebia christi there, and they published this nice paper just last year. And actually, this is the original area where the species was first described in the southern side of the Simplon Pass, so the Lagingtal in Switzerland. This is the border, this is the Swiss side, this is the Italian side. And it has become clear that the species, to start with, is much more widespread in the Italian side than was previously thought. So that was already an interesting discovery. But it goes further than that, and that is, on this map you can see in red are vertical transects, and as you saw on the photo, these are literally vertical transects. And they did a lot of research on those vertical mountains, and they found out that Erebia christi is actually quite widespread as far as they could see on these vertical transects, and much less so on the grass slopes below. And of course, this makes a difference. This is actually really the site we see on the left picture, but this is looking up, and this is always difficult. This is almost vertical, and this is just almost vertical, but very steep. As long as you search for Erebia christi on this side of the grass, you won't see many, almost none. There might be an occasional one coming down, but actually the species lives on these very steep, almost vertical rocks. So this paper, this research by 2 Italian researchers already show that once you really dive into it, the things can be different than we thought before. Of course, this also matters for the monitoring, because this graph has been shown by David Roy as well. And then we see that at the moment, monitoring data is especially available for the species which are actually considered least concerned. These are the widespread species, which can be found near every village or near every town. So all of you who do a transect somewhere in the neighborhood can come across these species. But for the rare and threatened species, there is only very limited data. So monitoring tells us something about how widespread species are doing, but it doesn't tell us all about the rare species. What we do know from those species, and this might be very interesting for the look into the future, is of course that some species show very clear trends. Argynnis paphia is a species which seems to be increasing in Europe. And actually also when we make a map with the trend per combination of butterfly monitoring scheme and by geographical region, we see that the species is actually increasing almost everywhere in Europe. Of course, we also have some species which are in decline. Beloria euphrosyna is a typical species which is not doing very well in most of Europe. But also, and that might be unexpected for some, a species like the small tortoiseshell, Aglaeus urticae, has been in decline on large parts of Europe. And actually the numbers are down by more than 70% now. Some show some mixed trends, like this Alatonia, the Queen of Spain fertility, went declined and then increased again, or Lysandra Belagus, the Adonis blue, which did the other way around, went up and down again. So things are not just straight lines. And also in the patterns on a European scale, we can see differences like this. This is Polymatus icarus, the common blue, and we can also see that it matters where you are. In some parts of Europe, the species is actually doing quite well. In other parts of Europe, the species is in decline. Also, the Article 17 reporting for the EU countries is a very useful tool to look at what has happened in the last 6 years. This was work done by Timothy Falls and Sue Collins, who collected the data, which is simply publicly available, actually, with the results from the Article 17 reporting. And this case is a very one of the real beautiful butterflies that we have in Europe, Panaceus Apollo, the Apollo. And what we can see already from this this overview is that this species is in a very bad conservation status, and actually only a few places where it's actually in a favorable conservation status. Most of them actually alpine areas, so the alpine areas of Poland, of Italy, and of Germany. These are the places where the species still is in a favorable condition. But in the rest of Europe, the species is actually doing really bad. In spite of a lot of efforts, this species is still declining and in an unfavorable conservation status. And also Matt already showed in his first talk today, that also in the US, in the western part of the US, many butterflies are actually declining. So it's a pattern we can see in many more areas. So when we look at how the butterflies are doing, we must say that some species are highly threatened, but the widespread species, the monitoring results show what we could call a mixed bag. There are some species increasing and some species declining. But for many of the rare species, we don't have real good counts, which is still a problem, or we simply don't know. Of course, expert opinion can help to bridge the gap, and that's what is allowed also in a red list assessment. So that can fill that gap, but it's still something we would very much like to move away from in the future, to have much better data on the rare species as well. So what are the threats for Europe, for butterflies in Europe? And actually, yesterday morning in the first talk by Michiel, we did this small poll, and I showed the results that we had. So most of you actually thought that agricultural intensification is the major threat. And it wondered me that abandonment is actually on place 5, where I personally think it's much more of a threat. So we also to talk about nitrogen deposition and the effects of that yesterday by Joost Vogels. I think that for many people that really was an eye-opener. Anyway, I was just interested in how you would feel about this now, after we have heard 2 days of talk. So we start again a polling, and you can all try to indicate which of the threats for butterflies in Europe, you can indicate the 3 most important ones according to you. And I'm just curious to see what happens, if it really is different from yesterday or that it's more or less the same. So we just wait a little bit until we have all the votes, 50 votes in now. Just wait a few seconds longer. There's a lot coming in still. We're all voting. Okay, I think that it stabilizes, so I end the polling. And we look at the results. And what you can see is that again, agricultural intensification is by far the most important, but it's interesting for me now to see that abandonment has clearly gone up and nitrogen deposition is considered a bit less important. But climate change is now number 2. So that was the same as yesterday. Thanks for doing this. Okay, we move on. Okay. It's also interesting to look at what we thought was 10 years ago during the last red list, the main threats to butterflies in Europe. And at that moment also intensification came out as the most important threat for butterflies, both threatened species and non-threatened species. Abandonment 2, climate change as number 3. So I wanted to focus on these 3 main drivers of change in Europe. Now, of course, we all know that we want grasslands to look like this, and actually this is what we all know for when we see this from a distance already, that there will be a lot of butterflies in these. But I showed this already before. This is what a lot of these grasslands nowadays looks like, at least in the Netherlands and in many parts of the Atlantic. And I come back to that specific matter later on. So what we see as a consequence of agricultural intensification is that all these areas that we here see, they are actually simply sterile. They're very close to green desert without any butterflies. The only butterflies you can find in these intense agricultural areas are restricted to road verges and nature reserves. So you have big fragmentation, small remnants left, but it even goes further because nitrogen deposition and water extraction lead to declining numbers of many butterflies. Netherlands and Flanders are probably the worst example of this. And what has happened, this is actually 3 maps. They are all of the same area, but then in different times, periods. This is all very close to the office of the Vlinderstichting in Wageningen. We are simply a little bit right below is where the office is. And originally this area was full of wet grasslands, just like the one I showed a few slides ago, full of orchids and with lots of butterflies, a very nice butterfly fauna of wet grasslands. Now, this was completely drained after the war with the Marshall help from the US, of course, given with very good intentions, but result was that this huge area in the middle was completely drained and changed into very intense grassland agriculture. Also, the photos I just showed you about the intense agriculture that have been made in this area. And all the butterflies actually just remained in a few small nature reserves you see back here. But after that, we still see that the monitoring, which started in 1919, shows us that after that, the butterfly numbers have gone down with over 50%. So even here you can see that where we have 5 butterflies here, we have 3 now. So the numbers are going down. But what you also can see, even in the Netherlands, which is a relatively rich country, look at how this nature reserve here actually is now half covered with woodland, relatively uninteresting woodland, because the typical butterflies of this area, typical rare plants, they're all typical species of hay meadows. So we've lost actually more than half of the area already to woodland. So that's how things go in an intense agricultural landscape. And of course, also the nitrogen deposition is typically a problem. The field to the right will be probably very heavily fertilized. And of course, it's typical that this blows over and infects all these grasslands to the left where the butterflies do live. And this nitrogen deposition also goes quite a distance, not about hundreds of kilometers, but up to easily up to 10 or 20 kilometers. And what you can also see is typically the Dutch fact that many of the nature reserves are actually don't have the money to mow all the grasslands, and parts of them get overgrown and turn into a wet forest, which is good for some species, especially Apatura iris can be quite abundant here, but still. We've seen also the effect, this is the slide that Thomas Fartman already showed about the host quality. So the effect of nitrogen is actually that the survival of the caterpillars goes down. A lot of nitrogen is not good, and Joost Vogels explained also how the rest of this effect worked. He also showed this that this effect of nitrogen deposition goes right into the heart of all the nature reserves in the Netherlands. In the Netherlands, we don't have any nature reserves or heathlands with a low nitrogen load. So we can only split between a high nitrogen load and a very high nitrogen load. And we can see that butterflies are butterflies, but also other organisms, but for butterflies, this effect is the same, are declining everywhere, but they are more declining on very heavy nitrogen load. So in intense agricultural areas, this is a serious problem. So it's also important to memorize that agricultural intensification is not restricted to agricultural land, because the nitrogen deposition and water extraction works to the middle of even our largest nature reserve. So butterflies decline even in those large reserves. And when we look at the overview of the results of the Dutch butterfly monitoring, and we summarize, we make a multi-species indicator of all the species, we see that actually this is split up into 3 periods. We see a decline in the first period, so up to 2004, a stable period up to 2013, and then it starts to decline again. With what we learn, we know that now this first decline was actually caused by the intensive agriculture and nitrogen deposition from that agricultural area into the nature reserves, which meant that also in protected areas, species were declining. Actually, that also was going on in the middle period, but then we saw that climate change more and more had a positive effect on some of the butterflies, but also that we saw the first signs that woodland butterflies were actually increasing, which is a phenomenon I come back to later. In the last period, we still have increasing numbers of woodland butterflies, but we see that the climate change effect is now turned negative, so it moved down. We still have intensification, we still have nitrogen, but the droughts of the last few years has been the driving force to let butterflies decline further. And what you can also see that since 1992, actually, we've lost now way over 50% of our butterflies, and we're on the way. Within 1 or 2 years, we will be at 60% decline. The other one is abandonment. I think that is, of course, a typical thing in many parts of Eastern and Southern Europe. I will shortly discuss the differences between intensification and abandonment. Of course, for the specialists in this type of areas, it's bad anyway, but you also notice there are still patches which are fine. So fields and meadows turn into secondary forest, but of course these small patches can remain okay for quite a while. It's also good to remember that this abandonment does not lead to nitrogen deposition into nature reserves, and also that the soil is not degraded after, because after intensification it can be really a tough job to get back to beautiful meadows again. So recovery from abandoned patches is relatively easier. We can also see that when we look at the results of the indicators that we built. This is the widespread indicator using all the species which occur. And what we can see is that actually in many areas, the last few years, species are increasing. So there are more species increasing than declining. But in the Atlantic zone, this is different. And one of the things might be that actually in the other areas of Europe, there is less intensification than in the Atlantic zone, which might cause butterflies to profit from the ongoing climate change. Whereas in Atlantic parts of Europe, we simply have so few butterflies left that a bit of climate change extra is already leading to real declines. Let's dive a little bit deeper into that. So these are the two main indicators that we've built during the ABLE project. Grassland butterfly indicator still showing a decline, but also the decline seems to be fastening in the last period again. It looks a bit like the Dutch. So first a period of decline, then a stable period, and then it seems to start to decline again. But the woodland butterflies, also on a European scale, they seem to be doing quite well in the last period. So climate change is one of the drivers behind this. So we go now to the third of the main drivers of changes in butterflies in Europe. It's important to remember that climate change is not just a nice warm springs, but also includes droughts in summer. Of course, we see the mobile generalist species moving north and the cool species getting isolated. And of course, this is a really spectacular example of Araschia levana, the map butterfly, moving north very far into Sweden and to Finland, and now already having reached the northern Norway, actually. And what's also important is that this recent paper, Michiel already talked about it, by Fourcade et al., shows the differences between dispersed and aggregated versus little and large amounts, and which showed actually that spatially dispersed semi-natural habitat favors the colonization of warm adapted and mobile species, and on the other hand, that the extinction risk of cold adapted species was lowest in aggregated areas. Of course, what abandonment means is that you get more of this, because abandoned landscapes, forests will grow, but there will remain small patches everywhere in the landscape for quite a while, at least the coming 10, 20 years. Whereas this is more the typical thing you get into intensive agricultural landscapes, is that a few nature reserves are well preserved, but in between you get completely empty areas. So what would be a small look through when we have seen all this knowledge? What could we expect for the future butterflies in Europe? So unless something really changes, agricultural intensification will move on wherever possible. And I also fear that outside the Atlantic zone, there will be a continuing abandonment of semi-natural grasslands. But climate change is now really coming in as one of the extra factors on this. So in the Atlantic zone, it's really expected that grassland butterflies will continue to decline, also because nitrogen deposition really goes into the heart of the nature reserves. But outside the Atlantic zone, I personally think that especially specialist grassland butterflies will be most affected by abandonment, because they need good areas of good quality, but many of the more generalist grassland butterflies will do quite well. Woodland butterflies will continue to increase, as we've already seen happening, especially outside the Atlantic, simply because there will be more woodland because of abandonment. But what we will also see is a further expansion of climate opportunists, which will, in theory, be faster outside the Atlantic zone, since the paper by Fourcade showed that small dispersed patches like you have in outside the Atlantic will be favorable for those species. Of course, we will also fear a further decline of the species preferring cool habitats, and that especially where the habitat is dispersed. And for that reason, I was really glad to finalize my talk with a slide from Frank Vassen. He showed in his talk, so that Europe is seeing the importance of having a good trans-European nature network, big enough to at least try to protect many of the land of the habitats for butterflies. And so I think this is a very good first step, and I hope this will, so my expectations for the future on butterflies, this will help to turn that into a positive way. So thanks for all. Of course, I will be there for questions, and I look forward to see all of you in a few years in real life. You can contact me over the email, but also on the websites of Vlinderstichting, BC Europe. And I think, I don't know if it's been mentioned already, but there is also the www.butterflymonitoring.net. And this is where you find all the results of the ABLE project and where you can join in monitoring wherever you are in Europe. Thank you.
Okay, thank you, Chris. I think this was a very nice wrap up of the symposium, and yes, of course, the outlook is well, is grim in many ways, but yes, Frank did open some perspective.
Yeah,
and actually there was a question on that because. Well, would an update of the Habitat Directive's list of protected species be an option to favor butterfly conservation or not?
Of course, the Habitat Directive uses the species to protect habitats, and that's, I think, the main purpose, and I think it is doing relatively well in that. Also, if we would change the species lists of the Habitat Directive, it would mean all the Natura 2000 areas would have to be reshuffled. Some might lose their status because they have it, because there is one of the species. They might you might get new areas. It would mean a lot of disturbance, I think. The main reason I would not favor a change of the Habitat Directive list is that it will open up also a complete new discussion. And I'm not sure if the forces that the agricultural community, having a much bigger lobby network and much higher funding, they will also try to reduce the effectiveness of the Habitat Directive. So I would prefer to count our blessings and to move on as we are, personally.
Thank you. Yes, I think we'll have to leave it at that for the questions, but we'll continue with the breakout sessions after closing this, and you can stay in there as long as you like. Take your lunch over there. But before that, I would like to close this this symposium, and I think we had a very nice session, and that is what at least our impression is, that it was really worthwhile to have us all together. I don't know how many unique visitors were there, but at least we had between 150 and 190 participants at one moment, so that was worthwhile, I think. Of course, we'll meet again in some time, and hopefully in a live setting again. We don't know the plans of Butterfly Conservation UK, who, well, should take over in organizing a European symposium. We assume that they will just organize it next spring, and that we will organize again a symposium in 3 years' time to catch up with the lost or the delayed symposium of this year. So, well, let's continue with the good work, and I hope you found the symposium inspiring. We will send you a link later on to the talks that you can watch back, and I wish you a safe trip home. Well, no, I guess it's not necessary, but so have a nice day, and see you later on somewhere. Okay, bye-bye.